Okay, so good evening, everyone. Welcome to this NPTEL live session. So I am going to be, I am the TA for this course, Cell Culture Technologies, and every Monday from 6 p.m. To 8 p.m. I take generally, but yes, last Monday I could not take. So today, because I was out of station, so I was not able to take the classes. Today I don't have much slides prepared because I was not I did not get the time. So maybe we will just have a discussion and then some questions we will solve. And if you have any doubts or any questions from your side, it will be more of a discussion session. So I have few slides. Maybe I will just go through those slides, and then maybe we'll have a short discussion and then solving the questions. Okay, so according to the cell culture lab guidelines, as we have been going through in the previous 2 weeks, so we had seen that cell culture lab needs to have a lot of rules and regulations that needs to be followed in order to avoid contamination, in order to avoid other issues. And there where we had seen that for in cell culture lab, generally people prefer for those who are doing dissections or those who are doing primary cell cultures, like isolating cells from animals. So people prefer to have a dissection hood or a area inside the cell culture, which is actually separate from the other normal secondary culture, like secondary cell culture hood. So separate area for dissections and cell culture hood is required for reducing contamination. So we had seen in earlier classes that if the cell culture room, in a cell culture be there should be corners should be placed in such a way that those are not the dissection area should not overlap with the cell culture hood area, like where you are normally doing the other cell culture work, in order to reduce the contamination. And also we should have a lot of precautions with using ethanol, using proper gloves and footwear, lab coat. Also another thing that is that most labs do nowadays to avoid contamination is to place sticky mats in front of their cell culture labs. So sticky mats help in removing all the dust particles from your foot while you are entering the cell culture facility. So it is it is now known that from dust particles and from air, there is a lot of contaminating microorganisms present in the air. So how much ever we are able to reduce, that is better. We cannot totally control that. We cannot have a totally contamination free or a micro free zone. That is very impossible to create. And if it is possible to create, it is very, very expensive. Like for example, the clean room setups, or for example the BSL-4 facilities where people are working with very, very contagious viruses or contagious bacteria. So there we need to have a very clean environment, but those are really expensive. But for normal cell culture, what we do generally is not very, very much sophisticated. But yes, we need to have certain precautions. We need to wear all those footwear, proper footwear, separate footwear. The footwear that we should wear inside the lab, inside the cell culture lab, should be different from what you are wearing outside. Also, some people also prefer to go barefoot. Like the cell culture lab should be totally clean, and people can go barefoot as well. So yeah, so we need to have dedicated footwear, lab coats, sticky mats, etc. To prevent contamination. All these are sources which can prevent contamination. Then we should have a refrigerator storage area. So last classes we did not much talk about refrigerator, but refrigerators are really very much important for cell culture storing the chemicals. All chemicals, including the media, including the transfection reagent, the plasmids that we transfect inside the cell, or say whatever other nutrients we want to give separately, or treatment we want to give to the cells, like some drug treatment or anything. So we need cell culture fridges, and those cell culture fridge should be separate from the fridge that we use in the normal laboratory, because cell culture items should be ideally separated from all the normal bench work items, and we should have dedicated refrigerator space for that. And refrigerators inside the cell culture room should be placed in such a way that it should not block the entrance, and also the door can be opened quite properly. So there are certain regulations as to where we should not place the hood. Like hood should not be placed, hood and refrigerator as well as incubator, because those three things are, it can be open, like incubator or hood, we are opening it. So it should not be placed in a direction where the window is there. So for example, we have if we have the cell culture rooms opening over here, and we should not and the air is coming from here, so just opposite to that we should not place any hood. That is a very, very wrong practice, because incubator or any cell culture hood, the air that is coming directly from the entrance, that will directly go to the this hood or this incubator, and can be a possible source of contamination. So also another source of contamination in the cell culture room are ACs. So ACs are kept like here. So AC air, if it is enters, because AC, this like they have filters to protect from these particles, and also if AC is not clean for a very long time, then it will have lot of dust particles. So AC air should also not enter the incubator when the incubator is being opened. So it is better to keep the AC also in a direction where the AC's air, like the fan is not moving in the direction where your incubator's door is opening. So these things are like some trivial things, like very simple things, but can cause a big problem. Like once contamination starts, it is very, very difficult to get rid of it, and it is very difficult to get like a proper contamination free area to work. Also, another important thing is the microscopes. This is the the heart of cell culture. We have to have proper good microscopes to see whether the cells are in proper morphology, whether the cells are healthy, or whether the cells do not have any contamination. So we definitely need microscopes, and it is better to have a fluorescence microscope inside the cell culture room. But if fluorescence microscope is not available, at least a bright field microscope is very, very important. So microscopes, for what reason? So cells generally, they tend to have a certain kind of morphology. So different cells have different morphology. Like some will have star-shaped morphology, some will have elongated morphology, some will have like some kind of like this kind of morphology. So depending upon what cell type we are choosing, it will be different. So depending upon what cells you are working with, your cell morphology is different, and depending on the morphology, we can identify whether the cells are correct, or whether the cells are healthy. So if the cells are healthy, all whatever the morphology of your cells are, for example, if the cells are this kind of star shaped, then there's the entire surface, or the entire boundary of the cells will be continuous like this. It is not going to have any broken ridges inside it.
Excuse
If it is,
me, ma'am.
yes.
Are you
Screen is
sharing
not
your
visible,
screen?
ma'am.
I mean, we can't see your screen if you are sharing it.
Oh, you're not able to see a screen? Okay,
No,
okay,
no,
one
since from
second.
the beginning.
Okay, one second, one second. Oh yeah, I forgot to share the screen. Okay. Now you're able to see, right?
Yes, ma'am.
Okay. So actually, I was talking about this, all these points I was discussing, that separate area for dissection, and dedicated footwear, lab coat, etc., refrigerator storage, and then we were talking about microscopes. So like microscopes, so basically, as I was talking, that we need it for studying proper cell morphology, or the whether the cells are healthy, or whether the cells are contaminated or not. So basically, when we see in the microscopic images, depending upon what cell type you are working on, the microscope, the cells morphology will be different. So it can be like star-shaped morphology, like some kind like this, or it can be elongated shape like this, or it can be like neuronal cells can be like very much fibrous in nature, like some like this fibrous nature like this. So you will see some fibrous nature of cells. So depending upon whatever cells you're working on, your morphology is going to be different. And by looking at the cells morphology, we can figure out whether the cells are healthy or not. So this is the nucleus. So suppose this cell is a healthy cell because it's the corners or the cell membrane is very much intact. But if the cells are not healthy, then we will see something like this. It's not going to be like a proper straight cells. It's going to be like this. So basically this cell and this cell, this is an unhealthy cell and this is a healthy cell. So by looking at bright field images, we can actually figure out whether the cells are healthy or not healthy. So if cells are stressed, we like not healthy means cells are stressed, and then cells may be stressed for various reasons. One reason could be that the cell culture incubator is not maintaining proper conditions, like it is not maintaining the carbon dioxide level, or it is not maintaining the temperature. That could be one reason. Another reason for cell getting stressed is it is contaminated. So contamination, when we are seeing it in microscope, it is very difficult to observe mycoplasma contamination because mycoplasmas are very small and we cannot detect it with our naked eyes. So we have to take the cell culture dishes and we have to do real-time PCR with mycoplasma genes, the genes which are present in mycoplasma, and we have to use primers against those genes so that if we are having mycoplasma, then those primers will bind to the mycoplasma genes and will get an amplification, and we can figure out whether there is mycoplasma contamination or not. Other than that, fungus, bacteria, and viruses. Viruses also, it is not very easy to detect it. Viruses also, for different kind of viruses, definitely if there is a virus contamination, cells are going to burst. Like this is not going to stay a whole. It's going to be like small fragments of cells. So if viruses are there in your cell culture, you will not see a cell as intact because virus will eat up bacteriophages basically. Sorry, not bacteriophages, like bacteriophages are viruses, but other kind of viruses also. So they will eat up the cells and they will take nutrition from the cells, and the cells will totally get fragmented or it will get burst. So virus contamination is like, we have to find it out like that, but we cannot detect viruses also with our naked eyes because those are also very small. What we can do detect is fungus and bacteria. So bacteria, different shapes are there. As you all know, there are coccus, like round shaped bacteria. There will be rod shaped bacteria, which is like rod in nature. And then there is like spiral shaped bacteria, which is spiral. So this kind of bacteria we can see in the cell culture, like microscope, where your cells will appear very big and your bacteria will appear like a small dot, like small dots you will see outside the cells. But also sometimes what happens is these dots should not be always, it is not necessary that these dots are going to be bacteria. It can also be that these dots are some kinds of these vesicles, because sometimes cells secrete some small vesicles. So those vesicles are also secreted inside the like cell culture dish, whatever dish you are culturing. But how to identify whether these are vesicles or whether these are contamination is, we can see that these small dots will be moving. Like whether if it is a bacteria, it will either move like in front, back, or it will show a vibration kind of movement. But if it is a particle, if it is a particle, it's not going to move. Like with your cell culture dish, if you are moving the cell culture dish, like the media is there in your cell culture dish, with that media it will show movement, like some particles. If you are putting some particles in suppose if you are putting sand in water and then checking it in a bottle, then you can see if you are moving the bottle, then you will see the particles moving. Otherwise it is mostly in rest, or maybe some little movement, not like a movement of spiral bacteria. Like spiral bacteria is going to spiral. Round shaped bacteria, they will also show movement like this. They will move. You can clearly make out which is moving, which bacteria, whether the bacteria is moving or not, or whether it is a vesicle that is secreted out from the cells. So vesicles are secret, like this is sometimes a characteristic of cells, that they secrete vesicles for their cell to cell communication, or some kind of like some chemicals they secrete that they put that is present in those vesicles. So finding out whether contamination is there or not, we have to use different kinds of microscopes and different kinds of objectives. Like higher magnification is better to see contamination, because this small bacteria and bacteria only generally. So this bacteria is not visible in lower magnification. We cannot see it in 10x, we cannot see it in 5x. We can see little bit in 20x, and better if we are seeing it in 40x. So depending upon like whatever your requirement is, so we can use different levels of microscopic objectives to observe not just contamination. Contamination is just one of the side things like which we should avoid, but real experiments we need to use a higher magnification objectives for seeing mitochondria, for seeing ER, etc. So microscopes, these are upright microscope and inverted microscope. So for example, in upright microscope, the objectives are in the upper side. Like we can see, these are the objectives basically. So these objectives are present in the upper side of your microscopic in the microscope. So when you are seeing it through your eyes, so this is the eyepiece, this is called the eyepiece, and here we can adjust it. This knob that you can see, this knob is where we can adjust. There could be two knobs, one is coarse adjustment and one is fine adjustment. So coarse adjustment is to focus it. Like sometimes when you will see that this objectives will go down, it will go down in this way. Like if we are coarse adjusting it, this objective is going to go down and going to go near your cells or near your plate where you have the cells. So one problem with the inverted microscope is it is very good for fixed samples. What is fixed samples and what is live samples? So if you have a glass slide and you have some cells here, and you have another cover slip on it, on top of it, and you have fixed it. Basically, you have fixed the cells in the sense those cells are not live anymore; these are dead cells, and you have fixed it with a cover slip. So this kind of imaging, or if you have a live, or if you have a fish, or if you have a insect like Drosophila or something, those kinds of insect, they are placed in some agar media, like some kind of soft agar media, just so that the animals don't move. So they will be placed, this is suppose an agar media, and in this agar media those kind of insects or fishes, they are placed over here. So this is suppose a fish, and so this fish now, when it is fixed in this agar containing petri dish, so this is not going to move anymore. So it's going to be here. So the upright microscope is very, very important for observing fishes or observing fixed samples which are thicker and which cannot be penetrated using an inverted microscope. So there are as you go deeper into microscopy, we can see that there are different kinds of objectives, different kinds of way how microscopes are used because of depending upon the requirements. Like for example, fishes, or for example fixed tissue samples, these are quite thick slices, and so if we are using the objective from the below, like from towards the inner part of the microscope, then the light will not be able to penetrate that deep inside the tissue or inside the live organism, that we can observe observe the phenomena what we are trying to observe inside that sample. So when we are having microscopes which are inverted, so light is getting reflected from here. So this light can be totally this light can pass through the thicker sections of the tissue or fish, and we can observe your whatever phenomena we want to observe. For example, if in fish, sometimes what we do is sometimes we image the fish brain. So we have the fish kept, like fish, not the fish but the embryo of the fish. So the small embryo of the fish, we put it in agar, and then we put it in this in this place, in this stage, and then we shine light from below. So the light is coming from below of the microscope, and with the objective we have to focus it. So for imaging like this, we don't need a better resolution. Our main purpose is not to get proper resolution. We don't want to see mitochondria, we don't want to see ER, we don't want to see other organelles. We just want to observe the brain, or we just want to observe maybe some kind of other organs or some development changes. If we want to see, like in embryo, we see observe the spinal cord, how it is getting, like how a small fish gets developed into a bigger fish, like from one cell stage, two cell stage to a bigger like not spinal cord they don't have, but the vertebral column. So yeah, so that all things we are using upright microscope to observe, because it is not possible to see those thicker samples inside in the inverted microscope. But what we do use generally in our lab, we use both kind of setup. We have upright microscope in our lab. We also have inverted microscope in our lab. So upright microscope generally we use for zebrafish samples, for studying the development of zebrafish embryo, for imaging the zebrafish brain. So those things. But what we very regularly use is the inverted microscope for cell culture. So here you can see that the objective is placed below, unlike the earlier one. Here the objective was on the top, and if we are focusing it, the objective will go down towards the sample, and here it has a chance to hit the sample. So those working distance has to be properly maintained, and those working distance needs to be standardized, like depending upon your requirement, because different microscope will have different objectives thickness or different objectives magnification, and it will differ from whatever sample you are using. Like for example, if some person is using tissue sample and some person is using some like fish sample, so their requirements will be like different. It is not going to be same. So it is very important to tell to the microscope manufacturing company what are your requirements, and accordingly they will customize the microscope working distance, because that is very, very important to have a proper view of whatever sample you are trying to image. So basically in inverted microscope also we have the objectives which are placed inside, like in the inner part of the microscope, and light is also shined from this part only. Light is shined from this part, so light is falling from here, and the objective is here. So here your objectives are placed. We can change the objectives on the basis of whatever magnification we want. We have 10x objective, we have 20x objective. There are different magnification is there. So 10x, 20x, 40x, whatever. So depending upon that, we need to, depending upon what we want to observe, we can change our magnification. And so there are two components of an inverted microscope. So if we have a fluorescence microscope, then generally this part of the microscope is important for fluorescence. This is called the epifluorescence part of the microscope. Okay, so this part of the microscope is important. And if we have, so this is called epifluorescence. So epifluorescence.
Ma'am, what is fluorescence microscope?
So fluorescence microscopes generally are used for seeing images which are having some GFP or RFP, like any kind of tag with it. So fluorescence microscope, what it does is, it will have, so here as I was saying, this is epifluorescence. That means that the light that we are shining, so for I will just write it, fluorescence. So in case of fluorescence microscope, the fluorescent part, like the light that we want to shine, suppose we want to get a green fluorescence light. If we want to observe a green fluorescence, then we have to excite it with a blue light. Always the principle of fluorescence says that whenever we are exciting with a light of a lower wavelength, then the emission will be at a higher wavelength. Okay, so there is always this some diagram that you might have seen, that if you are exciting some electrons, okay, these are some electrons and these are the energy levels. So if you are exciting these electrons at a particular energy level, so again it will come back to the original level, but it will not come back, so it will release some kind of energy, like it will release some energy while coming back, as heat or some other part, but it is not going to come back to the original level, but just one level above it. So we are going to get a wavelength which is high, like higher as compared to what we had excited it with. So because some energy is already lost while coming down. So suppose if you are exciting it here, like suppose this is the blue light's wavelength, and after exciting from here, so in case of fluorescence microscope, the light for fluorescence is from below, and the light for bright field or the light for white light, like if you want to see cells which are normal, like not fluorescent, just the cells, the bright light images, the bright light is focused from the top. So that is why I was saying that we have two parts in a fluorescence inverted microscope. The below part is epifluorescence, and the upper part is known as diafluorescence or something. So in the below part we have microscopic filters which can filter out different wavelengths of light. For example, if I was talking about to observe GFP tagged cells, then we have to excite with blue light. So what this microscope will do is there will be filters placed in the back side of the microscope. There will be different kinds of filters, and so if I give command to the microscope that I want to observe green light, then the filter which will obstruct all other lights and only allow the blue light to pass for exciting. So here only blue light is passing through this filter. So once the blue light is passing through this filter and all other lights are getting obstructed, then this light will come here and hit the surface of your cells which are placed over here. Once the blue light is hitting the surface of the cells, if your cells have any kind of fluorescent material tagged with it, GFP for example, then the GFP fluorescence will be like, it will get excited, the electrons in that level will get excited, and we can see the GFP fluorescence through this kind of eyepiece, or if it is connected to a camera in the in the screen, we can see, like if there is a monitor placed just next to it, we can we can observe that the green fluorescence through either eyepiece or through the monitor. So basically what that means is we are blocking all other sources of light and only exciting our sample with one particular source of light, green light, blue light, whatever light we want. And so whatever one wavelength above it we are observing. So for example, if you want to see red, then we have to excite it with a blue light. We have to excite it with if we have to yellow light or orange light. So like that. So depending upon whatever color your protein is tagged with, whether GFP, RFP or YFP. So GFP means green fluorescent protein, RFP means red fluorescent protein, YFP means yellow fluorescent protein. So whatever your protein is tagged with, one wavelength below we have to excite it. So if it is green, then we have to excite it with blue. Red, if you have to excite with orange, like that. So depending upon that, orange or yellow. So depending upon whatever fluorescence you want to observe, we can have different filters. Actually, the filters that we put in the microscope, these are very expensive. Like along with the objectives also, the filters are also very expensive. So depending upon what filter do we need, we have to also tell the microscopic person that we need blue filter, we need green filter. If we have to observe DAPI, DAPI is a stain which we use for observing nucleus inside the microscope. So if we need to see blue color, we need to have blue filter. So basically we'll need to have a lot of filters in order to see each colors separately. So those things, like those part of the microscope is responsible for like showing the fluorescent images. Like here, we will see a background as black and your cells are going to show green fluorescence or yellow fluorescence or whatever. But in case of bright field images, we are not going to see any kind of like black background. We will see like white background cells, and then we will see the cells, normal cells, like placed here. So that is the difference between your bright field and fluorescent microscopes. But generally microscopes, fluorescent microscopes are also bright field microscopes. Not all bright field microscopes are fluorescent microscopes, but all fluorescent microscopes are bright field microscopes, those. So bright field microscope, we don't have any people generally don't use bright field microscope for any experiments as purpose, just to see whether the cells are in proper morphology, just to see whether cells are not contaminated, cells are healthy. Those things bright field microscope is good. Also, sometimes what happens is bright field microscope can be used for seeing whether cells are differentiated or not. Suppose if we are working with neuronal cells, for example, if I work with neuronal cells, I work with neuronal cells. So neuronal cells, if it is in undifferentiated state, it is going to be like this. But if it is differentiated, it will show branching like this. So those things we can observe it in bright field microscope, and we can say that, okay, our treatment is working. Our cells which were earlier not differentiated, now it is getting differentiated because it is showing branches. Like neuronal cells tend to show like branches because of the axon fibers and all those things. So those things only we are seeing it with bright field microscope. Rest all other microscopic experiments that people generally do is with fluorescent microscopes, or now even better, there is confocal microscope. So confocal microscope helps in getting better resolution. So as compared to our upright microscope, the inverted microscope's resolution is better because we have a lot of filters, we have better objectives. In upright microscope, we don't have objectives which are 10x, 20x, 40x. I think the upright microscope objectives are 1x, 2x, some like very less resolution, because we are not working with like very small structures there. We are working with brain, we are working with an embryo, which is like very big structures as compared to cells which are very small, some micrometer in length. So inverted microscopes, there are advantages, disadvantages for every kind of system or every facility that they are using. So inverted microscope we cannot use for seeing bigger animals, or we cannot see for tissues. It is very difficult to image tissues. But for cells it is very nice and very like convenient option. And generally, like cell culture people, they will use inverted microscope in their like to observe cells. So next, we will talk about the objectives and numerical aperture. So what are placed inside the microscope, like objectives are very important to, like, it is basically that helps magnifying the image. But the image that we are seeing, the cells that we are seeing is originally not so big that we couldn't see it with our naked eyes, just because of the help of these objectives that we are seeing those images. And microscopy has advanced a lot in the recent years, and we have very good objectives. We have objectives like 20x, we have 10x, 40x, and even higher magnification like 60x or 100x even. So this kind of microscopes that are like higher magnifications, 100x or 60x, they cannot use just air as the medium for seeing the cells, because those objective lens that we have. So here, so this is basically how the objectives are placed. So this is the back side of the objective. Here you can see some screw like thing. So here we place it in this part. We just screw it, this kind of objectives, you can just screw it in the lower part of the microscope, and these are the lenses. This small part that you see, this is the lens of this microscope. And this lens has to be very carefully handled so that there are no like fungal growth, because what happens is, in our lab also we face this problem. If any spillage happens, suppose if you are imaging something in inverted microscope, if you are imaging something, you can see there is a hole, or there is a, if you are putting any sample there, if any spillage happens, if your cells containing buffer, if your cells containing any kind of liquid, if it falls on the surface of this objective, this inside the lens, there will be growth of fungus. There can be growth of any kind of not bacteria, but like fungus, or some kind of spores can be grown, and those are very bad, because when you are trying to image it, this lens, inside the lens, if these are growing, you will not be able to get a clear picture of your cells or clear picture of your sample, because the lens is contaminated now. And for cleaning this part of the lens, we have to call people from the company, and they charge a hefty amount to just clean the part of that lens, and also it takes a lot of time. So basically, microscopic experiments, we have to be very careful that we do not spill anything on the top of the, especially in the inverted microscopes, because if you are spilling anything, everything will go and fall on the objective, because objective is just below your sample. Objective is just below your sample. So here is your sample, here is your objective. So basically, your objective is just touching the sample, and anything, if it is spilling on the objective, and the lens are going to suffer a lot. So this lens, which is used for higher magnification like 60x or 100x, this lens is not compatible with air as the medium between the objective and the cells to observe like good resolution. This lens is compatible with some oil or some water. So in our lab, we have both compatible lenses, which is compatible to water as well as oil. So now, as I'm saying that compatibility with oil or water, what does it actually mean is, it is actually affecting the numerical aperture. So maybe you have heard this term, or maybe you have not, but what is numerical aperture is, so whenever you are putting any light source, okay, in any kind of system, so that light is entering that system. For example, this is the sample that we want to image, and between the light source and between the sample, there is some gap. There will always be a gap. So this gap is either filled with air, either filled with water, or glycerin or oil, or whatever you want to fill it with, like depending upon your requirements. But there is always some gap, and that has to be filled with any like component. Generally, for other other objectives, we do not use oil, do not use water for lower resolution. But for higher resolution, this air that is present in between the microscope, in between the light source of the microscope and the sample, the diffractive index is not enough to excite the cells, or to excite the molecules inside the sample. So the refractive index plays an important role in the in changing the numerical aperture, or the NA that we abbreviated with. So NA of the microscope. So numerical aperture, what does it mean? So this microscope objective, so it is shining light, like the light will be coming from bottom, and so for example, in inverted microscope, light is coming from the bottom, and so it will get it will be exciting the cells, and then it will get reflected through the objective, and your objective is going to then pass it to the camera or your eyepiece. So this immersion medium that you are seeing here, this immersion medium basically has a refractive index N, and that refractive index affects the reflection or refraction of the light that you are exciting it with. So this specimen, basically whatever light it is reflecting or refracting, that light is going to be affected by the refractive index, as we had seen earlier, like when I think during our high school, we have been taught that this refractive index is very important for our, refraction of light. So this refractive index is what matters when we are doing imaging of higher resolution, because if the refractive index is less, for example, in air it is less. So if this refractive index is less, then this, the light will not be reflected at a proper angle, and if it is not reflected at this proper angle, so this is the angle. So if it is not reflected at this proper angle, then it is not going to excite your cells, and you will not see any image. So you might think that whether your sample has problem, or whether the microscope has problem. So just to see whether your sample or microscope has problem, you will be confused, because if you don't see anything without by doing everything correctly, then you should actually apply oil, or you should see it with water, or see what objective you are using, whether it is compatible with air, whether it is compatible with water or air. Generally, in all the objectives, you will be be seeing whether it is compatible. For example, here you can see it is compatible with oil. So we have to we have to put oil on top of the lens. So here on top of the lens we have to put oil, 1 drop, very less amount of oil, some 3 to 4 microliter of oil we have to put on top of the lens, and then we have to image it to get the proper resolution. Otherwise we are not going to see any kind of images here, and then we will be wondering whether the problem is with the sample or not. So every objective, so it is very important to, whenever you are starting imaging or whenever microscopic experiments we are studying, it is very important to see what kind of objectives you are dealing with, whether the objective is compatible with oil, whether the objective is what resolution it is, whether it is sufficient to see what you want to see, whether it is sufficient to distinguish between the organelles or not. So everything is written basically. So there are other different kind of microscope companies, they prefer to write different things, but generally this oil compatibility, or whether it is compatible, certain objectives are also compatible with certain kind of light sources, like some will be compatible with fluorescence, some will not be compatible with fluorescence. So those things are also written, whether it is compatible with fluorescence, whether it is not compatible with fluorescence, whether it is compatible with phase contrast. So different kind of a wide range of microscopic techniques are there, which maybe someday we will see. But yeah, these are a lot of things to keep in mind while we are imaging the cells. So this is some fluorescent image that I have shown. This is basically my images that I am showing it here. So these are basically endoplasmic reticulum, you can see. Okay, so you can see this whenever you see fluorescent images, one thing that comes to mind is very beautiful images, because you will see colors, like in a black background you can see red color, yellow color, green color, whatever background you want to see. So these are some images where you can see endoplasmic reticulum. So here you can see that in the inside part of the, so this is a cell, and this is HEK293T cell, and this is how HEK293T cell looks like under fluorescence microscopy. If you remove the fluorescence microscopy, you will just see the outer boundary of the cell in bright field, so you will not going to see any color. So in fluorescent microscopy, you can see the cells are like this, and you can see some dotted structure inside it. You will see nucleus, all those. But in fluorescence, you will be able to distinguish whether it is in the ER, mitochondria, depending upon what kind of objective you are using. So in this fluorescent microscopy, we can see that here we have the nucleus that is not colored. So the nucleus is black color. You will see the nucleus is not colored, but the surrounding region of the nucleus is somehow in green color. But ER membrane, if I zoom the picture, then ER membrane generally forms a mesh-like structure. I don't know whether you are able to see it, but it is not going to be like a continuous structure. Depending, like it is going to be mesh-like. It's going to be basically like mesh kind of thing you will see. So it's going to be like this mesh region. So then you can understand that this is some endoplasmic reticulum you're imaging. So this is the endoplasmic reticulum that I had imaged from one control and overexpressed cells, and we can see that there is a increased in fluorescence intensity. So basically here what I had done is there is a so different fluorescent probes are present which can measure calcium levels, like the probe in the sense plasmid. So those plasmid has a calcium binding region, and just adjacent to the calcium binding region is the fluorescent region. So if the plasmid binds to your calcium, and if it fluoresces, that means your calcium is present inside the cell. So higher fluorescence means higher calcium is present, lower fluorescence means lower calcium is present. So here is one example of an experiment that people do, is to how people use fluorescence microscopies to check whether the calcium levels are altered or not, or whether sometimes when people check with different dyes to see whether cell death has happened or not. So if cell death, there are different staining procedures, annexin PI staining or FDAPI staining. So like cell death. If it has happened, it will show red color. If it has not happened, it will show green color. So like that. So it is very interesting technique to use in different kinds of experiment, and it is very like it is very enjoyable technique also, I feel personally, to image the cells and show like beautiful colorful images. So yes, this is an ER calcium, and here I have shown one video. So this is live calcium imaging that I had done. So if you see this GIF, I think you can see the levels. You can see that in this image it will go down and again go up. So go down, go up. So these are time lapse images basically. So apart from just taking snapshots or pictures, even some microscopes are capable of taking time lapse images. Okay, so depending upon your requirements, obviously not all labs require time lapse images, because some labs are okay with showing this the snapshot, not like over the period of time what is happening. But in our lab we are very much concerned with over the time, whether the calcium is decreasing, increasing, not changing, whatever. So we need time lapse imaging, and so this is one example of time lapse imaging. So this is over a period of 3 minutes, 4 minutes, you have to image particular dishes or samples and check whether your calcium levels are affected or not. So here these are again ER calcium levels, which is decreasing or increasing. You can see that the time lapse images. So this kind of this is another example of. So this is not so here also you can see these are yellow colored cells, but this is not to measure calcium, it is to measure chloride. And in chloride levels, basically we have like in ER chloride, the sensor is yellow color, so the cells are in yellow. So here one thing I want to mention is the cells which are rounded. So you can see some rounded cells over here. So this rounded cell, not this. So this rounded, like where the fluorescence intensity is not like gradient. Like for example, in this cell, you can see here nucleus is not colored, and again in the side we have yellow color. But in this cell, this cells, whole cell is yellow. So these cells are actually dead cells, and these cells so in death, when cells are dying, so the plasmid is totally accumulated everywhere inside the cell. So these cells are not considered for like publication or for analysis. But the other cells where you can see this kind of like healthy cells, so here you can make out what are healthy cells, what are not healthy cells. So healthy cells will look like this. It will look in morphology, it will not show this rounded kind of morphology. If cells are getting rounded or if cells are floating, then it is going to be like dead. So basically so imaging is also necessary for seeing whether your cells are proper or whether your imaging is happening properly or not. So this is about fluorescent imaging, and yes, definitely it's a very interesting technique. And cell culture, it is highly people who are working on cell culture, and they generally require a lot of microscopic training and microscopic exposure to like see beautiful images. So apart from that, one topic I wanted to discuss is clinostat. So in our lab, we don't have it, but this is a very sophisticated device. So basically what happens is in the incubator, so the incubators, not yesterday, last week I had shown the incubator image. So in the incubator, we have 5% carbon dioxide and we have 37 degree Celsius temperature we are maintaining. But we don't have cells which are but we have racks, like we have rack in which we put the cells. We don't have a moving incubator which is going to like neglect the gravitational forces. So definitely in our incubator, all the gravitational forces are applying, and we have cells, we keep it in the rack, and all the gravitational forces do apply on the cells. And I don't know whether any physiologically affect, so definitely it's not very physiologically similar to what our body has, because we are continuously moving and we are sitting, getting up, so it is, we are under motion. But when cells we are growing it in the incubator, it's totally stagnant. It's in a plate and it's totally stagnant. So clinostat is a device which like people who want to study exactly and duplicate exactly the physiological condition, and who cannot afford a little bit error also, so they use this device, and this device is used to control the effect of gravitational pull by rotating. So generally this device is like a kind of some disc kind of thing which will continuously rotate and will reduce the effect of gravitational pull. It will not totally avoid it, because it will still you will still experience the cells will still experience gravitational pull, but it will definitely reduce the effect. So many people place this instrument inside the incubator and put the cells on top of it, so that your cells are in motion, and it is not totally stagnant, and it's not going to settle. Like for example, if we are putting any particles, sand in water, and if you keep it for some longer time, you'll see the sand settles down. So it's not going to happen like that. But if the cells are adherent, definitely it's going to settle down. And that will be more physiologically relevant, that it is not because of your like gravitational pull your cells have settled down, but it is because of its characteristic that the cells have settled down. So people use this kind of sophisticated instruments for their studies. But yeah, these are very like I think very highly physiologically relevant, those who want to study, so they kind of use it. And the advantages are definitely, they have low shear stress and active diffusion. So whatever media we are growing in it, and whatever like treatment we want to give, so it's properly spread in all part of the dish, and it's not going to settle down in one part because of the gravitational pull. And also cells are being more physiologically, like they will show more physiologically relevant changes that are like similar to in vivo conditions. So yeah, so this is clinostat, and this is like, yeah, people use it, and but we don't. So yes, so I have slides still here only today, because I said I was not actually available. I had gone out, and so yeah, now we'll discuss some questions, and then before that, if anybody has doubts, maybe we can discuss those. Also, I want to really apologize because I have not uploaded the video, and I have also not uploaded the slides, because I was really busy with the, I had this conference, so I was really busy, and I just came 2 days back. So I will upload it. So I'm really sorry, but yeah, I will do upload it. Anybody has any questions, then we can move into the question answer part.
And what is the difference between polycarbonate plastic and the polystyrene plastic?
So depending upon like, so there can be coating. Basically, lot of cells, they are not, they quickly, so lot of adherent cells also. For example, in our case, we have HEK293T cells. So although they are very adherent, but if they are so polystyrene and polycarbonate, depending upon their substrate, like depending upon their like composition, they help in proper adhering of the cells. So 293T, for example, HK293T, for example, it is adherent cells, but if you wash it very rigorously, or if we not rigorously also, if we wash it very gently also, cells tend to move away. So polystyrene or polycarbonate coating, they help in properly adhering the cells. Like though they have chemicals that can bind with the cells, like adhesine, or I don't adhesine or some other like molecules, which will allow the cells to keep adhered in the surface of the dish, so that you can image them. Because if the cells are very loosely attached, then if you are giving a wash or giving a media change, then the cells tend to float and move away. So you will not be able to see any cells because cells have moved away. But this coating helps in providing ample amount of like restriction, so that the cells don't move, and that is because of its like interaction with the cells, the adhesive molecules, like that. So those are like some chemicals that people have over the period of time, they have optimized that this chemical helps in proper adhering of the cells. So normally all the cells, like neuronal cells, these are very rigid cells, they will not move. Also there are these cancer cells, certain cancer cells, those are very rigid cells, they don't move. But certain cells, like 293T and all, they are very fragile and they are loosely attached. So for those kind of cells, we need to have this kind of coating.
Ma'am, why polycarbonate flask is autoclavable and why a polystyrene flask is not autoclavable?
Because like not autoclavable in the sense that those are chemicals, right? So if we are autoclaving it under pressure or under such temperature, so most likely those chemicals might get degraded. So for which the purpose was actually generally cell culture, all the things that we use for cell culture is one time use. We should not autoclave it. And only like if there is like very much emergency, then only we should autoclave the things we used for cell culture. Otherwise it is highly recommended not to use like reuse the things, or autoclave it and reuse. But yeah, to answer the question, that if you are autoclaving those chemicals, then maybe it is getting lot of chemicals, not only just polystyrene plates, but there are certain chemicals that we use in cell culture. For example, BSA, bovine serum albumin. It is not autoclavable because it's a protein, and if you autoclave it, it will aggregate. So there are a lot of chemicals like that which are not autoclavable, so we have to filter it. So either we have to filter it, we have to UV sterilize it. So we have to find out other ways of sterilizing it, but not autoclave it, because it can get degraded, it can get aggregated, and it will lose its function for what it was made. So yes, but for cell culture, generally it is advisable not to reuse any kind of consumables, because once you are taking it out of the cell culture room, which is supposed to be clean, and it's under like a lot of contaminating substances, so it might get contaminated. So very high chances of getting contamination even after autoclaving also. It is not advisable actually. Yeah. Okay, so shall we move to the questions? Let's move to the questions. So yeah, today we don't have a lot of things to discuss, so we will just move to the questions. And again, I have to meet you on, okay. Should we use polystyrene flask only once, or are there ways to make it sterile and usable? So see, polystyrene coated flasks we get. Also, we get polystyrene in bottle. So if you want to use it, because if you want to use it in flasks, those will be expensive. And we should not reuse it. Generally, we should not reuse it. But if you are having bottle, we can always use chemical from that bottle and use normal flask that we have or normal dish that we have, and coating is nothing, just that will be in liquid form, and we have to just put 1 ml of 1 ml of it and like swirl it and keep it. That is the coating. So if you are if you want to reuse it, I would advise it. I don't think so we should reuse it, but if you are getting the bottle, it will be very helpful for you, because whenever you want to use it, then you can use the normal flasks and just coat it with the polystyrene and you are good to go. So it's like it's less expensive solution to that problem. Okay, so let us now go into question answers. So yes, so while using microscopes, if the magnification is changed to maintain the right amount of light, what do we need? So, okay, Ragul. So you have given option B as the correct answer. Actually, no. Why to maintain? So if you are changing the magnification lens, just the magnification lens, it is not the magnification lens is not directly affecting the brightness. I mean, light intensity is completely independent of what magnification you are using. So magnification, numerical aperture, and the refractive index, they are kind of like related to each other, but the light intensity is definitely a different part of the microscope. So your whatever light intensity you are using is not going to affect what magnification you're using. If you are using magnification of 40x, 60x for bright, this is for bright light actually, so it is not going to like affect like the brightness. So basically, to maintain the right amount of light, if you are maintaining the right amount of light, we have to like, in order to like 10x to 20x, what happens is we are increasing the resolution. So it means that the lens are now becoming more finer, like more thinner. So we have to, in order that the light should enter the cells, it is like it is not, sorry, I was saying it is independent, it is not independent. In order to, like, for example, for magnification, if it is increasing from 10x to 20x, that means the light, the lens is becoming thinner because the resolution has to improve. So the refractive index is maintained the same. We are not changing the refractive index. Only what we can change is the light's this thing, light's intensity. So the alpha that we are talking about, n sine alpha, so that has to basically, if that lens is becoming thinner, so in order to get more light into the cells, we have to increase the brightness of the cells, increase the brightness of the microscope, not decrease the brightness, because we are moving the magnification from lower to higher. And if you are moving it from lower to higher, more magnification means more thinner the lens is, so more amount of light is required to enter the cells. So we have to increase the brightness. It's not decrease the brightness, or it depends on the aperture of lens. So aperture of lens is depending upon several factors, right? Aperture of lens depend upon refractive index. It depends upon the angle at which the light is entering. So yeah, so aperture of lens will not so like aperture of lens is going to be not just a single entity, but it is made up of two different entities that it depends upon. So it depends upon refractive index, also it depends upon the light angle. So yes, so if you are increasing the magnification, always you have to increase the brightness of the light. If you are decreasing the magnification, decrease the brightness of the light. Also there is another term that is called exposure. So exposure means how much time you have to expose the cells in order to get that how much time you have to expose the cells in order to get good amount of fluorescence. Exposure time is used generally for fluorescent microscopes. So if we want to like see some cells which are very dim or which is not very bright, like not very actively bright, not showing actively bright fluorescence, then we have to increase the exposure time in order to observe that phenomena, or observe that fluorescence. So this generally happens in many cases, like where your expression level of that protein is very less, and in order to detect it, we have to increase the exposure time, like for the amount of time at which the cells are being exposed to that sort of light. So then we have lens becomes thicker at x20. I think it becomes thinner because we have to so it becomes thinner or thicker, I don't know, but it becomes more concave, I guess, more concave. If we have to see a better resolution, the lens have to become more like concave, like this, like concave part. So it has to become more concave. And as it increases, as the magnification increases, so as the magnification increases, we have to use a better refractive index in order to counteract this part. So aperture comes down at x20. Numerical aperture is n sine alpha. So if your aperture comes down at, whether it will come down, it's not I physical aperture. Physical. What do you mean by physical aperture? I'm not sure what do you mean by physical aperture, or whether x20 is smaller because of shorter f. In the sense that focal length or something you're saying. Focal length. Yeah, I'm not very much into the physics of the lens part, but I don't know, focal length, aperture. I am not very much well versed with it. And I think somebody had asked a question last time that how oxygen had supplied, like how oxygen is supplied in incubator. So in incubator already we have, so 5% carbon dioxide is maintained using the carbon dioxide cylinder, but inside the incubator we are not. So inside the incubator we already have normal air. Like we don't have. So when we open the incubator, when we close the incubator, we don't have any other like other system to maintain different air. So whatever air we have in the room, that air is there in the incubator as well. Just 5% carbon dioxide additionally we are maintaining. So oxygen is already there in the incubator itself. Okay. So yes, and apart from that also the water that we are using, maybe that water also gets some like dissociated or something from where we can get oxygen. So yeah, oxygen inside the incubator is already present. Like just in the normal air we have oxygen, so we have air there. Okay, so next question, let's go. What type of microscope is needed to observe cells written on a piece of metal? So piece of metal is a thicker thing, so what do we need? Like if cells are plated on plastic or cells are plated on thing, then we need a like other kind of microscope. So yeah, so the correct answer is option B, because metal, you know, it's a piece of metal. Metal is a thicker substance. So cells, if it is placed on metal, we need to have an upright microscope to observe it. So hi Pavani. Final exam, I'm not sure. It should be objective only, but I'm not sure. I cannot so I'm not in the part of like conducting exams, so I'm not sure whether it is objective or descriptive. But I think it is objective only. I'm not sure, but okay. Yeah. Yes. So most of you have given the correct answer. So if magnification of the lens is increased, means if it is going higher magnification from 10x 20x, so basically your lens is becoming more concave, and your so the distance that you require between the lens and your cells or specimen, it will get reduced, because it is like coming in more closer contact to the specimen. The lens is coming in more closer contact to the specimen. So magnification, if it is increased, then the distance or the working distance specifically means the distance between the specimen and the lens, so the objective basically. So the working distance means the distance between the objective and the specimen. So if the magnification is increased, then the working distance gets reduced. The next question. Correct. So yeah, the correct answer is clinostat, as I was discussing. So in order to maintain negation to reduce the effect of gravity, it's the zero gravity simulator, clinostat. So it helps in maintaining a very much physiological conditions in the cells, and by continuously rotating. So the next question. Revant, we are not preparing question paper. We are just like TA for like live sessions. There are other TAs who are like for the course, like who are maintaining the NPTEL page and the question answers. So we are not in the team for like conducting exams or those. Just we are for the doubt clearing sessions. So we will not be preparing question paper. Yeah. Yes, so everybody has given the correct answer. So it's option A. Sticky mats should be placed at the front entry gate of the lab. So in order to, while entering the lab, we should properly, all the dust particles in our food should be cleaned. Actually, this question, I also have a doubt, as in, like, the answer for this question is C, but actually it should also be B. Like, in vitro studies, not only for like glass vessel, we also do it in plastic dishes as well. So it is not like just C. Like, the option is not. I don't think the option C is correct. Both B and C should be correct, because it can be done on glass vessel. It can also be done on plastic vessels. So yes. So both B and C should be correct. The next question. Yes, so the correct answer is C. It's not A. It's just not 0.5%, but 5%. So 5% carbon dioxide levels are required. Like, it maintains physiological conditions inside the cell culture incubator. So yeah, option C is correct. Yes, so correct answer is option D. So T75 means 75 centimeter square surface area of the flask. So option D is correct. The last question. Okay, so the correct answer is option A. That's right. So, any other questions for today? Okay, 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 Kaushik. Thank you. I did not know that. Okay. Okay, so if there is no other questions, then maybe we'll say bye-bye and see you again on Monday. So for fourth week's lecture. Yeah. So thank you everyone for joining. And see you again on Monday. Bye.